All right, in this video, we'll be looking at part 2 of tonicity and osmoregulation, specifically focusing on this concept of water potential. All right, so water potential is measures the tendency of water to move by osmosis. You can think of it as the potential for water to do something, if you want to think of it that way. And so there's a particular equation for this. It has to do with both pressure, because pressure obviously can affect the movement of water, and it has to do with solute, because as we learned in the last video, the amount of solute in a solution or on one side of the membrane can cause water to move into or out of that area, because of the water, or the amount of solute causes water to move toward if there's more solute, or away if there's less solute. And so that's where we get this equation, whereas you have total water potential is equal to the pressure potential, and pressure can be negative or positive, plus the solute potential, and the solute potential is going to be a negative number most of the time. I don't know that I've ever seen a positive solute potential. And so you can have a zero solute potential if there's no solute, obviously, and you can have a zero pressure potential if there's no pressure. You could have a total water potential of zero if there's no pressure or no solute. And so, but again, as we're dealing with this, you're going to be comparing two systems in this. And so a couple rules to know. Water moves from an area of high water potential to an area of low water potential. And so you can have negative or positive values for total water potential. The more negative the water potential is, the more likely that water will move into that area, which makes sense. The unit for this calculation is going to just be bars, is the unit for it. And again, your pure water has a water potential of zero as long as it's in an open container. Open container represents no pressure, and pure water also has no solute. So this idea of osmoregulation, organisms control their internal solute concentration through different means, and that's what we mean by osmoregulation, is an organism controlling its internal solute concentration. Increasing the solute concentration on the inside will increase the water potential, and will decrease the water potential, and cause water to want to move in. And so you can do the opposite. You can increase by changing the pressure, by changing the solute. And so organisms have different ways in order to regulate this. There are certain systems that regulate the amount of solute, like kidneys are an example of this, or there are other ones that can regulate pressure. Some organisms have systems that actually push water out of the body. And so depending on the need there, so in our own bodies, kidneys can detect the amount of solute in the blood and can adjust accordingly, causing you to get rid of excess water or to store water depending on solute concentrations, because there is a particular set value that the body needs those solute concentrations to be at, and the kidney will recognize that. More complex organisms like ourselves have very complicated kidneys. Fish have less complicated kidneys, and they live in water, so that brings this whole other set of problems to that. In order to calculate total water potential, you have to be able to calculate solute potential. And so there is a particular equation for that. You can see that there. The solute potential is equal to the ionization constant times the molar concentration times the pressure constant times the temperature in Kelvin. I is called the ionization constant. And basically, the question you can ask yourself is, how many pieces is this going to turn into if I put this in water? So the number of particles molecule makes in water. So something like sodium chloride, that water is going to ionize into sodium and into chloride. Whereas something like glucose, which is an organic molecule, it's going to just have one particle. It's not going to separate. This will almost always, I've only ever seen it be one or two. One if it's a sugar, and 2 if it's some sort of salt or acid or a base. That is what I have seen. Molar concentration is going to be a given. You're not going to have to solve for molar concentration. This pressure constant is always going to be given to you. It never changes, 0.831. And temperature, most likely you will be asked to convert Celsius into Kelvin, and you do that by adding 273. Solute concentration or solute potential is always a negative number. And here's how you know. See that little negative right there? It's the problem that students make most of the time is that they forget to multiply that by -1. Obviously, if you have no concentration, pressure, or temperature, then you're multiplying 0 by that -1, which is 0. So it is possible to have 0 to be your solute potential.